Hello everyone, welcome to NEET Buddy. Today we are going to provide you with 15 MCQs from the topic that you're seeing on your screen. So the practice of these MCQs will give you confidence and you would be able to score better in the coming NEET exams. So without wasting much time, let's get started. Question number 1. First cloned animal is option A, dog, option B, cat, option C, sheep, or option D, goat. Which was the first animal to be cloned by humans? The correct answer is option C, sheep. Question 2. The carrier of hereditary material are chromosomes, genes, gametes, or gametocutes. The carrier of hereditary materials are. The correct answer is option A, that is chromosomes. Chromosomes carry the hereditary material. Question 3. Agrobacterium tumefaciens used in genetic engineering for option A, DNA mapping, option B, DNA modification, option C, as a vector DNA, or option D, DNA fingerprinting. The correct answer is option C, vector DNA. Question 4. Any changes in gene frequency in a population is called as. Any changes in gene frequency in a population is called as. Option A, gene flow, option B, genetic drift, option C, gene pool, or option D, gene conservation. The correct answer is option B, that is genetic drift. Question 5. Recombinant DNA technology is related with option A, Darwin, option B, Stanley Cohen, option C, Herbert Boyer, or option D, both B and C. Recombinant DNA technology is related with which of the following scientists? The correct answer is option D, that is both B and C. Stanley Cohen and Herbert Boyer are related to recombinant DNA technology. Question 6. Technique for production of monoclonal antibodies was initially developed by option A, Stewards Skoog, option B, Lister and Koch, option C, Arber and Haberland, option D, Kohler and Milstein. Technique of production of monoclonal antibodies was initially developed by. The correct answer is option D, that is Kohler and Milstein. This technique of production of monoclonal antibodies is also known as, yes, hybridoma technology. Question 7. A genetically engineered bacteria used for clearing oil spills is option A, Escherichia coli, option B, Bacillus subtilis, option C, Agrobacterium tumefaciens, or option D, Pseudomonas putida. The correct answer is option D, that is Pseudomonas putida has the function of clearing the oil spills, and it is a genetically modified or genetically engineered bacteria. Question 8. A bacteriophage is a option A, virus that parasitizes a bacteria, option B, bacterium with a lysed membrane, option C, P.PPLO, or option D, mycoplasma infecting bacteria. The correct answer is option A, that is virus that parasitizes a bacteria is called as bacteriophage. Question 9. A genetically manipulated organism containing in its genome one or more inserted gene of another species is called as. Genetically manipulated organism containing one or more inserted gene of another species is called as option A, transposons, option B, gene expression, option C, transgenic organism, or option D, retroposons. The correct answer is option C, that is transgenic organism. Question 10. Use of transgenic plants as biological factories for production of specialty chemicals is called as? Option A, molecular farming. Option B, molecular genetics. Option C, molecular mapping. Or option D, dry farming. Use of transgenic plants as a biological factory for production of certain special chemicals is called as? The correct answer is option A, that is molecular farming. Question number 11. Which vector is commonly used in transfer of genes in a crop plant? Option A, plasmid of Bacillus subtilis. Option B, Bacteriophages. Option C, Ti-plasmid of Agrobacterium. Option D, E.Coli Phages. The correct answer is option C, that is Ti-plasmid of Agrobacterium tumefaciens is used as a vector when you have to carry out a transfer of gene in crop plants. Question number 12. Structure involved in genetic engineering is? Option A, plastid. Option B, plasmid. Option C, codon. Or option D, none of these. Structure involved in genetic engineering is? The correct answer is option B, that is plasmid. And what exactly is plasmid? It is an extra nuclear chromosomal material which can be used as a vector to transfer genes in genetic engineering. Question 13. Chimeric DNA is? Option A, DNA which contains uracil. Option B, DNA synthesized from RNA. Option C, recombinant DNA. Or option D, DNA which contains only a single strand. Chimeric DNA is? The correct answer is option C, recombinant DNA is also called as chimeric DNA. Question 14. Genetic engineering aims at? Option A, destroying wild gene. Option B, preserving wild gene. Option C, curing human diseases by introducing new genes. Or option D, all of the above. The correct answer is option C. With genetic engineering, we can cure human diseases by introducing new genes in place of the genes that have been mutated. Question 15. Gene which is responsible for synthesis of a polypeptide chain is called as? Gene responsible for synthesis of polypeptide chain is called as? Option A, promoter gene. Option B, structural gene. Option C, rectangular gene. Or option D, operator gene. The correct answer is option B, structural gene. So those are the MCQs for today. We'll see you guys again tomorrow with another set of MCQs at this very place, this very time, from the next chapter of class 12th NCERT Biology. We are doing the daily biology MCQ test series over here, where we are providing you MCQs every day from one of the chapters of class 11th or class 12th NCERT Biology, so that you get a lot of practice before the coming NEET exam. So we'll see you tomorrow. Till then, all the best and keep working hard. Do like and share this video if you find it helpful, and if you're new out here, don't forget to subscribe to our channel. Thank you guys for watching. Bye bye.